Hi guys, today we'll be looking at the operon concept, that is, understanding what is an operon, and we will be focusing on the Lac Operon, which is used by the E. coli to metabolize lactose, which is a carbohydrate. So an operon is a functional unit of DNA. By functional unit, we understand that it codes for mRNA through the process of transcription, and then the mRNA can form one or more proteins through translation, that is in accordance with the central dogma. Central dogma. So it's a functional unit of DNA containing a cluster of genes under the control of a single promoter. That means that it's a regulatory mechanism. It's a regulatory mechanism. In prokaryotes. E. coli is a bacteria, and it's a method of transcriptional control. It regulates the formation of mRNA itself. Now, there are two ways in which an operon can work. There is positive control and there is negative control. The control is positive and negative. In positive control, there is a molecule known as the activator, which has to be present to form the mRNA. However, in negative control, there is something called the repressor. And in the presence of the repressor, transcription is inhibited. This brings us to the Lac Operon in Escherichia coli, commonly known as E. coli. Under normal circumstances, E. coli prefers to use glucose as its primary metabolite, that is, the carbohydrate used to metabolize. However, when concentrations of glucose are low, concentration of glucose are low, glucose, forgive the spelling mistake. E. coli can use lactose to metabolize. This is done by a system which is governed by the Lac Operon. So now imagine this is a sequence of DNA, which is the Lac Operon. There is a Lac-I gene, which is known as the regulatory gene, which is transcribed continuously. Like every other gene, it needs a promoter to enable transcription. The promoter is a place where the sigma protein, which initiates transcription, and RNA polymerase, eukaryotes have only one type of RNA polymerase. Prokaryotes have only one type of RNA polymerase, eukaryotes have three. And when both of these binds, they form an holoenzyme, which enables transcription. Now, the Lac-I, which is a regulatory gene and transcribed continuously, forms its mRNA, which codes for the repressor protein through translation. Repressor protein. When there is enough glucose, the repressor protein goes and binds to the operator of the Lac Operon. So when the repressor protein binds to the operator of the Lac Operon, transcription of the operon is inhibited. The Lac Operon itself also has its own promoter, which facilitates the binding of sigma protein and RNA polymerase, to again form a holoenzyme. In the presence of lactose, so lactose binds to the repressor protein. So this means lactose is the inducer, because when it binds to the repressor protein, it changes the configuration of this protein and it is unable to bind to the operator. Thus, when lactose is present, the repressor protein cannot bind to the operator, therefore promoting transcription. This is how it is negatively controlled. Now, the three genes present in the Lac Operon are LAC-Z, LAC-Y, and LAC-A. LAC-Z codes for beta-galactosidase, which cleaves lactose. That is, aids the metabolization of lactose. LAC-Y codes for galactosidase, permease, which enables the transport of lactose into the cell. The last is LAC-A, which codes for trans-acetylase, which exports the sugar from the cell. That is, maintains concentration of sugars by transferring the acetyl group. By transferring the acetyl group. Thus, when lactose is present in the surroundings, it binds to the repressor protein, thus enabling transcription of LAC-Z, LAC-Y, and LAC-A. However, this is not it. We mentioned that when concentrations of glucose are sufficient, there is some effect on the Lac Operon. This is because of two hypotheses. One is known as the inducer exclusion. Inducer exclusion. According to this, if glucose concentrations are high, glucose is accepted into the cell more easily. Thus, lactose does not enter the cell. The second mechanism that is proposed is under positive control. Control of CAP, which is Catabolite, activator protein. CAP binds to cyclic AMP, commonly known as CAMP, which binds to the promoter of the Lac Operon. Of Lac Operon. And when the concentration of glucose is high, CAMP concentrations will reduce. Thus, the CAMP-CAP complex won't bind to the Lac Operon. This reduces the transcription rate. However, when glucose concentrations are low, cyclic AMP concentrations are high. Therefore, transcription increases. To conclude, we have one challenging question. Let's say if the operator of the Lac Operon is moved ahead of the promoter, that is, if the operator is taken and moved here, what effect will it have on the Lac Operon? Think about it and comment in the comment section. I will let you know the answer in the next video. Hi guys, thanks a lot for watching the video. If you have any concerns, comments, feedback, or you want to ask anything, want new videos on something, do let me know in the comment section and I'll be happy to get back to you. Thanks a lot once again.